as we all know that most of the reports are saying that coronavirus has actually originated from a bat from a live market from Wuhan. So this is what our current understanding is. So how do we know that it has been originated from a bat is the first question. And why bat is a very good source of harboring viruses. Now, in the previously in previous instances also, you have this bat, which is a natural reservoir. So bat is a natural reservoir of viruses. This cartoon is actually depicting that the bat is hosting a dinner to various viruses. So the bat actually is a reservoir of various viruses, like previously reported SARS, that is severe acute respiratory syndrome. You have Ebola. You have MERS, that is Middle East respiratory syndrome, and you have Nipah. These are very dangerous viruses, and in the past they have been transmitted to us from the bats. So probably the scientists probably are now thinking that even the coronavirus has also been originated from the live market of Wuhan, which has been transmitted to us from a bat, because bats are very good natural reservoirs of viruses. So in that sense, the coronavirus is also been originated from a bat. But the first question which comes to our mind is, why is bat a good natural reservoir? So the answer lies here. Now, bat has a very high metabolic rate. Now, bat has a very high metabolic rate, and due to which there are harmful substances which are created in the body known as oxygen free radicals. Now, these free radicals are very harmful to the bat, and therefore they have to be eliminated. So if something alarming is happening in our body, then there has to be an alarm. So the alarm is inflammation. Sorry. So the alarm is inflammation. So something harmful is happening to our body or the bat's body. There has to be an alarm which has to be raised, a kind of siren. And that siren or alarm is inflammation. Inflammation means it is a secretion of certain molecules. Here in this case, it is interferon B and TNF alpha, that is tumor necrosis factor alpha and interferon B. These are the alarming factors. That means they raise the siren that there is a potential toxic substance in the body. And now when this siren is raised, then there are certain cells which are recruited to clear this toxin. These cells are nothing but T cells and B cells. So in case of bats, there is a very interesting and modified mechanism. And that mechanism is, it has a compromised inflammation system. That means even though bats has toxic substances in its body, the inflammation will not be high. That means the siren is not heard. When the siren is not heard, the police cannot enter the scene. So in that sense, the interferons as well as TNF alphas are secreted in low levels. So when there is no siren, it is always likely and possible that the toxic substances can happily harbor in the body of the bat. So in this case, the viruses also, that's the reason why, because of this compromised inflammatory system, the viruses happily can enter into the bat and replicate in the bat. Now the first target is over. The virus can actually sit in the bat happily. But the second aspect of this is the virus should not infect the bat. If it is infecting the bat would die, and therefore it cannot be transmitted to us. So the first process is, by reducing its inflammatory potential, the virus is in the bat. And secondly, it also has certain unique antiviral immune responses. Say, for example, this virus is replicating and it tries to infect the bat. Then there are certain molecules which are produced by the bat to combat the virus. So two aspects here. One, virus can happily harbor in the bat. Two, and it will not cause any infection to the bat. So our first question of, okay, bat can be a natural reservoir of viruses, and therefore this can be the origin of coronavirus. Now, how do we know this? How do we know that the bat is the source? So what scientists have done is they have isolated genomes of the person who is infected. A person, it is in December 1st, I guess. December 1st, they detected a patient with coronavirus in China, and they have isolated this viral strain. They have isolated the genomic DNA of the virus, and they compared it with the available database. So what did the database say? Don't get worried with all these pictures. This is just a phylogenetic tree which compares the closeness of DNAs. Now this is, I hope everybody can see this arrow. This is the SARS COVID, that is the SARS coronavirus 2. This is the novel virus. Now they have seen that the DNA is very much similar. You see this here, very much similar to a bat known as Rhinolophus affinis. Rhinolophus affinis. Now this is showing almost greater than 90% similarity to what was isolated in a human being. So that means, so the similarity between the patient, the similarity between the coronavirus isolated from the patient and the bat is almost greater than 90%. So obviously one would infer that whatever source of coronavirus would be from a bat. But interestingly, as many of the reports say that it is originated from bat and it is from China, it is not exactly originated from bat, because the first patient who was under whose lungs the coronavirus was detected was never exposed to seafood market. That means he never went to that seafood market. And when that patient has never gone to the seafood market, he wouldn't have been exposed to bat virus. So there are two contradictions here. One, you are seeing similarity between bat DNA as well as the isolate from the human. That says that, okay, probably the origin is from the bat. Two, if that is the case, this person would have been exposed to a bat, but this person was never exposed to that market. Then how did this virus actually enter humans from bat? So the question first is, okay, bat is a good natural reservoir. Bat can harbor viruses. Bat can transmit coronavirus. This is known. Now, the reports also say the bat DNA and human DNA, the virus DNA has greater than 90% match. So therefore, bat is also could be the source. But the irony is the patient has never been exposed to bat. So what could be the source? So as you see here in this picture, you have a bat here, which is the natural reservoir of virus. This is us, that is the human beings. Now there are some intermediate hosts. If you just go back to the history, you have this case of a camel, which is an intermediate host of MERS, that is Middle East Respiratory Syndrome coronavirus. Now this is also a coronavirus. And the SARS coronavirus, which is a previous version, you have an intermediate host, which is a civet cat. And it is established that through bat, the virus has been transmitted to this civet cat, and through civet cat, it has been transmitted to the human. Similarly, the bat has transmitted the virus to the camel, and the camels have transmitted the virus to the human in case of these two coronaviruses. But what about this novel coronavirus which we are talking about? Is it directly from the bat? So the studies are saying, okay, 90% match between human virus and the bat. But there is something, a missing link here, that there is some other intermediate host. So the question comes, why do you require this intermediate host? So you imagine a big overhead water tank in a colony. So the water is stored naturally there. So that is your natural reservoir, the bat. Now the water has to be circulated in the colony, and you imagine these intermediate hosts as the water tankers. So that means this is the natural reservoir which is hosting or harboring all the viruses. And these viruses are now transmitted via some connect, because humans are not directly in contact with bats. So therefore, there should be some animal in between which would transmit the virus, and due course of time, the studies now indicate that a pangolin is the intermediate host which could have carried the coronavirus into the human. So the first question which we are addressing is, what is the origin of coronavirus, whether it is a bat directly to the human, or whether it is a pangolin directly to the human, or whether it is a bat to the pangolin to the human. So now the studies most likely are reporting that a pangolin is indeed transmitting it to the human. Now, how do we know this, that a pangolin is doing this? So the second story is about pangolin here. So we have heard the bat story, and the second story is the pangolin story. So as we discussed, you have this MERS coronavirus, SARS coronavirus, and you have these intermediary hosts. Now the intermediate host of the virus which has been transmitted from bat could be pangolin. Now why is this possible? Because during the same time of this pandemic, they have also found in the lungs of one or two wild dead pandemics which are in close association with human colonies. They have found a similar coronavirus too when they have isolated the strain and they have isolated the DNA. Now what they have done, they have matched the DNA of coronavirus 2 found in this pangolin, and they have matched the DNA with the coronavirus 2 in the humans. And what do they find? So the results are quite interesting in the sense that, okay, you see that this is the human being and this is the bat, and therefore we have the coronavirus of human and the coronavirus of bat are very similar. Okay, and you also see a pangolin here. Now this is when you are comparing the whole DNA of human with whole DNA of bat with whole DNA of pangolin. But what they observed was, there are in due course of my lecture, I will discuss about these receptors. There are certain receptors in our respiratory tract known as ACE2. These are the receptors to which the coronavirus actually binds. A part of coronavirus binds to the ACE2, and that is how the coronavirus is infecting us. Now they have compared the ACE2 receptor of the pangolin as well as the human. And to their surprise, you can see that whatever sequence is there, the core sequence of ACE2 is matching with pangolin, not with bat. So they have come to a conclusion that probably, yes, bat is the natural reservoir, and the virus has mutated in pangolin, and the mutated virus is now transmitted to the human. So our next question we have addressed is, okay, the natural reservoir is bat. From the bat, it has been mutated to the pangolin, and from the pangolin, it has now come to the human. Now, this is a brief introduction about how it has been originated from a bat or a pangolin. These are based on scientific studies, and all the credits are actually given in the slides. So let us now focus on the coronavirus as such. So why are we calling this coronavirus as a novel coronavirus and not similar to the previous coronaviruses? Now, if you go just to the classification of this virus, it belongs to order Nidovirales, family Coronaviridae, and under Coronaviridae, you have two subfamilies, Coronavirinae, and you have 5 genuses, that is alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. So the previous 2 instances of coronavirus which we have are SARS coronavirus and MERS coronavirus, that is the Middle East ones, and the severe acute respiratory syndrome ones, coronaviruses. Now these 2 are the coronaviruses which are already reported, and we know the sequences also. Now this is something here, you see this is the SARS, we are naming it as SARS coronavirus 2. The name of the coronavirus, which is a beta form of coronavirus, is SARS coronavirus 2. That means it is different from this, it is different from this. We will also discuss how it is different from this as well as the previous ones. Now, the terminology should be very clear here. SARS coronavirus 2 is the virus, and the disease caused by this virus is known as COVID-19, that is coronavirus disease, which is detected in 2019 year. So COVID-19 and SARS coronavirus 2. So if you just examine this slide, there are so many coronaviruses in the past. This coronavirus is new, but this is not the first time that humans are exposed to coronavirus. In fact, we have 7 coronaviruses, including the present one, which we are exposed to. You can see that the SARS as well as MERS are also coronaviruses, and there are very other normal and common forms of coronaviruses. That means we were already exposed to coronaviruses. But what is so special about this? What is so interesting about this, that why are we focused more on this, and why we have actually not bothered to work on the previous coronaviruses and the MERS coronaviruses or whatever coronaviruses which are known to infect humans? So the answer is lying in this structure. So this is also an animation which has been obtained from Scientific Animations. So if you examine the structure of the coronavirus, now you have these spike-like particles. This is the main protein. This is the spike-like, which is the frame, the red frame which you can see is the envelope. And you see under this frame, there are some proteins embedded in it. And one such protein is spike protein, which is very, very important in terms of understanding how coronavirus actually works in our body or enters into our body and does its job. And you also have another protein SM. M is the M protein. H is the hemagglutinin protein. This hemagglutinin protein is also normally found in influenza viruses also. So you also have S, M, H, and E. You have E protein. Okay, so this is the envelope, and you have 4 different types of proteins embedded in it. One is the spike protein, the second one is the M protein, the third one is the H protein, and the fourth one is the E protein. And inside this structure, you have the genetic material of this RNA, which is the single-stranded RNA. Now, this is how you can visualize a coronavirus. It doesn't look exactly like this, but this is how, when you have to imagine a coronavirus, it looks something like this. And why it is very important is the transmissibility of this virus. Transmissibility means how fast it can actually infect another person. Say if today an X person has a coronavirus, and if it is infecting 3 people by tomorrow, then you say that in a particular term known as R naught, that is transmissibility. That means it can fastly transmit from, it can infect from one person to 3 persons very rapidly. Now that is the rate of transmissibility of this coronavirus, which previous coronaviruses doesn't have. The previous coronaviruses transmissibility is only 1 to approximately 1.5. But this particular virus has the transmissibility. That means it can spread fast. Mind you, this is not lethal. That means it will not kill the person until unless it is going to an advanced stage or with comorbid conditions. But it is spreading very fast. The spreading is given by R naught value. Now, a deeper look at this spike protein now, because this is the portion which goes and binds to the cells you have under this respiratory tract. Whenever you breathe, right from the nose till the lungs, you have a respiratory tract which is lined up with a particular cell known as epithelial cell. Now what happens here is, this is the spike protein. Now this spike protein goes and binds to a receptor which is present on the human epithelial cell. So let us just examine this spike protein more closely. So if you see the entire genome, so if I'm just going back, this is the genetic material, which is the RNA, that is the ribose nucleic acid. This is the genetic material, which is 29,903 nucleotides. Now this is the total size of the genome of the coronavirus. In this, you have a small portion which codes for this spike protein. It is marked in pink color. You see this? So this spike protein, again, if you are zooming in, you see this spike protein has various domains. This is N-terminal domain. RBD is known as receptor binding domain. This is the domain to which our receptors, human receptors, bind to the spike protein. Then you also have a FP, that is fusion peptide, IFP, HR1, HR2, and transmembrane domain. Now this is the spike protein structure. If you are going into the depth of the spike protein structure, you see that this is made up of 1, 2, 3, 4, 5, 6 domains. So this is, if you have to visualize the spike protein, you see this. This is the membrane. This is the membrane of the virus. You have this 1, 2, 3, 4, 5 main. Okay, so this is the S1 subunit. You see this? This is in the form of a balloon, three spikes. Now this is the portion known as S1. So from here to here, it is S1. Following which you have 1, 2, 3, 4 domains. These are the 4 domains. 1, 2, 3, 4. The 4 domains will become your S2. These 4 domains are S2. This whole domain is S1. And in the back of it, you have the transmembrane domain. So you have S2, S1. This is how the spike protein looks like. Now, why I'm emphasizing this spike protein among all the proteins, we have discussed about 4 or 5 proteins which are on the membrane of the virus, is because this is the protein which actually goes and binds or recognizes the epithelial cells. So as we discussed, why this SARS-CoV virus is important is, right now the transmissibility is 2.5. You see, the previous SARS coronavirus was 2.5. It actually reached more than 3 or 4 in certain countries, especially in India and America, and there are places, Brazil, there are places where the transmissibility is very, very high because of the high dense population. You have the transmissibility more than 3. So this transmissibility is what is worrying in case of SARS coronavirus 2. The second thing is incubation period. Incubation period is longer. You see this, the incubation period in case of the previous SARS coronavirus is less. Now, what is the significance of this incubation period is, the longer the incubation period, the longer the virus is in our body, but we are not able to detect it. That means you do not see the symptoms during the incubation period, but the virus is already there. So within this 4 to 12 days, it is very, very easy to transmit this virus from one person to another person. In this case, the incubation period is low. That means the person will develop symptoms faster. So it is easy to isolate this person, but this is difficult to isolate a person in a very short span. Therefore, the transmissibility is high. So I'm not going into the deeper details of this, but these two are the aspects which are very much important to understand why this SARS-CoV coronavirus. But again, one good factor, one positive factor is the mild illness is high in case of SARS coronavirus 2. That means majority of the cases you don't find death or very severe cases. You only find very mild or even asymptomatic cases. And you see the proportion of patients getting hospitalized are very less, that is 20%. So there are certain, what do you say, positive things with respect to SARS coronavirus 2 is that we need not actually panic that it is lethal, but it is highly transmissible. Now, just coming back to the structure of this coronavirus. Okay, now these are the spikes. Now, I just told you these spikes are being recognized by certain receptors on your respiratory tract. Now, this is your respiratory tract. And in the respiratory tract, you have two different membranes. One is known as angiotensin converting enzyme 2, that is ACE2. This is the receptor. And the other receptor, which we know is TMPRSS2, that is protease serine 2. These two are the receptors which go and bind, or which the coronavirus, whenever it enters through a sneeze or through droplet or through hand contact, and that hand actually moving into your respiratory surfaces, the coronavirus now sticks onto your nasal surface epithelial cells. How does it stick? The spike protein recognizes 2 receptor as well as the transmembrane protein serine receptors. So now after this recognition, what happens? I'll come to this slide later. So after this receptor binds to the surface, there can be two ways by which a virus can actually enter into your cell. One is known as the fusion pathway, and the second one is known as the endosomal pathway. So the virus, you see here in the endosomal pathway, the main molecule which is responsible is ACE2, that is the receptor, angiotensin converting enzyme receptor, which is responsible for the endosomal pathway. That means the virus as such, but this is the spike protein. The spike protein binds to the receptor, and this is been now taken as such, as an endosome, okay, into the cell. Now, this is one way a virus can enter. The virus can enter in the second way by utilizing both the receptors. Now, I just mentioned another receptor which is known as TMPRSS2. Now, this receptor also can bind and fuse to these spike proteins. And what happens is, you see the difference between these two pathways is only the genetic material, that is the RNA, is entering in case of fusion pathway. In this case, the virus is actually entering here. So there are two pathways, the endocytic pathway and the fusion pathway. Now, this is the virus entry. So after entry of the virus, what happens? I will just discuss briefly, and then I will come to the drugs which we are using to target each and everything. So this is the coronavirus here, and you have the spike protein. You have this ACE2 receptor as well as TMPRSS2. Now these two are the receptors, and one is responsible for endosomal pathway. The other one is responsible for fusion pathway. Now what happens here is the coronavirus now binds to this, and either of these pathways, either it can enter, only inject the genetic material, or it can enter as a virus into our cell. Now this blue one is our cell. This is the respiratory cell. It can be a lung cell, or it can be a cell in the epithelial cell in the nose. Now what is happening here is, in the endosomal pathway, there are various other molecules which are now cleaving this virus and removing this genetic material out. Now, as soon as what is the genetic material of SARS coronavirus 2 is single-stranded RNA. Now, single-stranded RNA is out. Now, if you go to the basics of molecular biology, a DNA is transcribed into RNA, and RNA is translated into a protein. Now, what is happening here is the mRNA, the RNA which is entering into the cell immediately uses the host. Host means humans. Immediately utilizes the host proteins to synthesize two different proteins. One is two different very big proteins. These are PP1AB and PP1A. These are two proteins which are corona proteins which are synthesized in our body. And this is now, with the help of two different proteins, another proteins will be cleaved into pieces. You see here, this is one protein. If you just take it as one large protein and one short protein, this is now again fragmented into smaller pieces, 1, 2, 3, 4, 5, 6. Now, all these are nothing but the components of the virus. The components of the virus means the proteins which we have discussed, the proteins of membrane, the proteins. So you have these large protein and small protein, which is further cleaved into smaller proteins, which will now in a way give rise to what you known as give rise to the whole viral structure again. Now, just going back, what are the available treatments, drug treatments? So disinfectants has been exhaustively covered, and now this is the drug. Disinfectant is something which we are using it not to get contracted by coronavirus. But say if a patient has coronavirus and now he has to be treated. Now you find three types of patients. One, asymptomatic. That means you really don't know that he really has any symptoms. But if you go and get himself tested because somebody else is tested, and he might be positive. Second, you have patients with mild symptoms, and these are the majority. Mild symptoms means you might have a cough, you might have a small fever, and there might be some loss of smell and certain other things. And the third case, which is very rare, is patients with severe corona symptoms, where you might require the support of ventilator as well as oxygen. So that is very less, that is only 10 to 20% of population. Now again, one more point the scientists have observed is it is very much prominent in the age group of older age. That means the older age people are more susceptible, and males are more susceptible than females, and children are less susceptible. Children are less susceptible. So these are some positive takeaways from the coronavirus. So let us talk about the drugs which are in underuse. One, you can block the entry of virus into the cell. Okay, so we discussed about two ways the virus can enter the cell. What are the two ways? The two ways are via the receptor, spike protein binding to ACE2 or TMB-RSS. They are the two pathways. So if you block the virus to enter into the cell first, that itself will take care of that. So the drug which is used there is hydroxychloroquine. This is generally a drug which is used in cases of malaria. For treatment of malaria, it is very much widely used. And it is a very cheap drug and available in generic forms also. However, now these drugs are known as repurposing drugs. Repurposing drugs means these drugs were earlier used for some other purpose. They are purposed basically for malaria, but they are showing some effectiveness for virus. So these are known as repurposing drugs. So one medication which is used commonly is hydroxychloroquine. What hydroxychloroquine does is it blocks the release of the virus here. Now this is the virus which is in a vesicle known as endosome. Okay. Now the endosome is highly acidic in nature. The pH of the endosome is acidic. Now by adding hydroxychloroquine, which is a base, it increases the pH of this endosome, and hence the release of virus into the cell is blocked. Now this is the hydroxychloroquine which is used. The second one which is used is which blocks the TMP-RSS. Remember, there are two different receptors. So the second one is camostat, mesylate, or one more is nefamtostatin. These two are similar role. That means they are blocking TMP-RSS. Now one can think why they are given in combination. Why they are given in combination? Because if you are blocking this pathway, the virus can actually enter into this pathway. If you are blocking this pathway, the virus can actually enter through this pathway. So if you are giving a drug in combination, hydroxychloroquine and camostat, the virus can effectively not enter into the cell. Now, this is one drug treatment which is widely used and reported across countries. No, I'm not getting into more details there. So initially you have chloroquine or hydroxychloroquine, nafamostat and chemostat, which are blocking the entry. Now these are known as entry blockers. Now suppose the virus has already entered. Still you can block the virus. Now how is that? We just discussed there are one large protein and one small proteins which are synthesized by using our host machinery by the virus. Now what they have to do is, now these proteins have to first degrade into smaller products to get assembled into a new virus. So what happens is these are proteases synthesized by the virus. One is 3CL-Pro and PL-Pro. These are proteases synthesized by the virus, which cleaves this large protein and small protein into smaller subunits. Now say if you are blocking these proteases. If you block these proteases, what would happen? This large protein and small protein will not be cleaved into individual components, and these individual components cannot be packed into a mature virus again. So in that sense, here you are using again 3 different drugs to block these proteases. One is lopinavir and ritonavir. Now these 2 are the drugs which block the protease 3CL-Pro, and the other one is disulfiram, which blocks the PL protease. Now, when you block these proteases, you are not getting the individual components, and hence the virus is not able to assemble into a mature virus and not leave the cell. So that means a virus can enter the cell but cannot leave the cell. Now, the third line of drugs which are being used is, and there are so many drugs actually which are coming up on a daily basis. But whichever are more efficient and whichever are widely or popularly reported, only I am trying to discuss them. And the third line of thing is remdesivir and favipiravir. These are the most popular ones which are actually being used. Now, what are they doing? Now, you see that there are some small components here. Now, these components together will form a particular complex known as RNA polymerase. Now, this polymerase enzyme is very much important for replication of the virus. The replication of virus is done by this enzyme known as RDRP, that is RNA polymerase. This is an RNA polymerase. Now, this polymerase is blocked. Now, for example, let us suppose at this step also we have failed, and the virus is able to synthesize all its components necessary for a virus. Now, the third step which we can block is replication complex. The replication complex is remdesivir and favipiravir, which goes and blocks and does not allow the RNA to replicate. Now, what happens when the RNA is not replicating? The genetic material cannot be packed. Even though you have, see, in a virus, you always have 2 structures. One is the capsid, the protein, and the internal one, which is the nucleus. That is the nucleic acid. So even though you have a outer head, you don't have any brain in it. So if you have a head and if you don't have a brain, that's of no use. So the brain is the RNA polymerase here. And therefore, when you are giving this remdesivir and favipiravir, the RNA polymerase is not replicating the genetic material. So even though you have a physical virus, it will not have genetic material to infect. So this is the third step. You are blocking virus at various stages. 1 at the entry, 2 at the cleavage stage, that is the proteolytic stage, 3 at the replicative stage. Now the fourth one is, okay, in spite of this, the virus is clever enough to actually bypass all these things and still replicate and form more number of viruses. Now once you do this, the virus is released. See, the more number of viruses are released because the virus is able to happily sit in this and replicate. Now, there are another 2 different types of drugs here used. One are interferons, which are antiviral drugs. That means the virus is there, it is replicating, more number of viruses are replicating. But if you are giving these interferons beta, they are antiviral in nature, which cleaves the virus. And there are other drugs which are known as immunosuppressants. Immunosuppressant means, as the name suggests, they suppress the immune system. Now, one might ask why you are giving immune suppressants, because it will suppress the immunity of our body, and therefore we will lose the ability to fight the virus. But here the catch is, as I discussed earlier, inflammation is always good. Inflammation means it's a kind of siren. When you have a siren, then therefore you have T cells and B cells. But if the inflammation is hyper, that means if there is a lot of secretion of these molecules such as interferons and TNF alpha, they can also damage our own organs. Although they act on virus, but they also damage our own organs. In that sense, you will also see that our lungs get damaged. This is in very cases which are very rare, in the sense that they need ventilator support, their lungs are damaged at this stage. That means the virus has entered, the virus has proteolytically cleaved, the virus has replicated, the virus has formed a very mature thing, and then it is moving out. At this stage also, you can actually administer these drugs. So these are and if you see, there are these are the list. This PowerPoint will be shared anyways later. So there are various drugs which are now being used across various countries and at various phases. Now I'll discuss about shortly. I'll discuss the last part of my lecture, which is vaccines. So these are the drugs and how these drugs act on the virus, coronavirus molecule. So we discussed about how it has originated, how it is transmitted, the structure of coronavirus in brief, and the drugs which are used and how they are targeting the coronavirus in our body. So the final aspect of the part of this presentation is the types of vaccines. Now there is a lot of urgency as well as a race which is being created among countries to develop a vaccine. But honestly speaking, to develop a vaccine, it will actually take, if you are following stringent pipelines, it actually will take anywhere between 5 to 10 years. Now, the first case of coronavirus was reported in December 2019. So, if you are imagining that the kind of pace with which the countries are doing, is that we can expect something positive by next year. So, we are actually curbing, the scientists are curbing approximately 6 to 7 years. And there are various strategies to prepare a vaccine. I will discuss 4 or 5 very important, and I will discuss what are the prominent vaccines which are in various phases. When you actually prepare a vaccine, it involves various steps. And once you know these steps, we will appreciate how cumbersome or how difficult this vaccine preparation is. So first of all, the phase is exploratory phase. Exploratory phase is where in the laboratory scientists would do various researches. They come up with some kind of breakthroughs. And based on these breakthroughs, they will identify certain molecules which would combat, or they would identify various ways to combat the virus. Now they have to first immediately cannot test these findings inside a human. So they have to use it in preclinical studies. In preclinical studies, you use animal models, that is mice. You have various mammals, primates, that is chimpanzees, guinea pigs. And there are various other models on which the vaccine has to be tested first. So let us suppose the preclinical stage is successful. That means it successful. When you call a vaccine is successful, is first it can elicit a very good immune response in the body. And immune response means, okay, raising antibodies and raising T cells. Let us put it this way, B cells and T cells. And this immune response should be persistent. That means if the next time also the virus will come, it has to remember that and it has to neutralize it. So first, to elicit a good immune response immediately, and to memorize this immune response for the future. 3, safety. That means it should not cause any kind of side effects inside the human. Efficacy, how effective it is in treating the patients. Again, if you are talking about how effective it is how effective is it in kids, how effective is it in patients of other diseases, that means who is having diabetes, who is having a cardiac issue, or whoever their things. So there are various stages at which everything has to be monitored. The first one is exploratory, the second one is preclinical in animal models other than humans, the third one is phase one. Phase one is where you actually pick up a small pool of humans in laboratory conditions, that means in hospitals. 20 to 80 patients or 20 to 100 patients are picked up which are healthy, and they are given a vaccine. I'll discuss what are these vaccines, but let me explain you the process of preparing a vaccine. So once this phase is successful, again successful means it has to raise good immune response, and that immune response, again if you are giving the same dose or higher dose, it has to combat the virus. 3, there should be no side effects, and the human beings are again healthy and happy. Now if this is successful, then you enter into the phase 2 process. Under phase 3 process, you will target various groups. In the first process, you will only examine very healthy volunteers, but in the phase 2, you will have to examine various age groups. That means you have to pick up certain kids, you have to pick up young people, you have to pick up middle-aged people, you have to pick up old people, you have to pick up all kinds of samples, and then you again test the drug for its safety and efficacy. That is phase 2. So even if this is also successful, then you go to phase 3. Now phase 3 is a very large level clinical trial which has to be done approximately 2,000 to 3,000 people. Okay, and now this is being tested in open. Mind you, the first 2 phases, phase 1 and phase 2, are in the laboratory condition, but this particular phase 3 will be now the patient will be administered the vaccine and he is going outside. That means how the environment is interacting, how he is interacting with other people, and it has been given to 3,000 more than individuals, and you have to see what are the complications. So after monitoring each and every patient who has been given this vaccine, this is the longest phase and the toughest phase actually in vaccine preparation. Once phase 3 is successful, now it is ready to be released for approval. So there are various bodies, every country has a body to regulate and approve it. So they approve it and then they are released into the market. Even after releasing into the market, it is still under scrutiny in phase 4. Like, okay, after certain uses, what is happening, whether the vaccine is effective or not. So these are the various steps involved, and this whole process actually takes around 5 to 10 years in a laboratory, in a very well sophisticated and well established laboratory. So one can appreciate the pace with which the countries are trying to prepare this vaccine. Now, vaccines are of various types. One is the whole inactivated. That means you will inactivate the whole virus. Okay, so you will inactivate this whole virus. Like in this case, if you are inactivating the whole virus, that means you take the coronavirus, you inactivate it with various, like you add some heat or something and you just inactivate it. That means it's not live. Now this inactivated virus is injected into your body to raise antibodies or T cells and B cells. Now this is known as whole inactivated virus. Now one such example which is being carried out of whole inactivated virus of coronavirus is from Bharat Biotech. The Bharat Biotech of India is using this whole inactivated virus. That means it is trying to inject it into the humans and trying to see whether this can be a good vaccine. One such example. The second one is known as live attenuated. Live attenuated means the vaccine is live. That means the organism is live here. The virus is live, but it is attenuated. That means it is weak. How you are making this weak? You are actually changing some parameters in this particular virus. That means removing a spike protein or X protein, Y protein. You are making this protein weak, but it is live. Now you inject this live but weak organism into the body. You call it as live attenuated vaccine. Now one such example which is carried out is Indian Immunologicals of India Indian Immunologicals Limited, and in collaboration with Griffith University, is trying to prepare a live attenuated vaccine. And another example is Codagenics and Serum Institute of India. The Serum Institute of India, in collaboration with Codagenics, is trying to prepare a live attenuated vaccine for coronavirus. So we discussed about whole inactivated, live inactivated. Now the third one is subunit vaccines. That means you will not pick up the whole virus. That means whole virus or live weak virus, but you will pick up a part of the virus. So we discussed spike protein is very important. So what they are doing is they will try to take this protein, isolate this protein, engineer it. I'm not going into the details of how they do it, but superficially you will just engineer this protein in such a way and inject it into the patients or individuals and see how this is eliciting the kind of immune response. So these are known as subunit vaccines. So one example of the subunit vaccine is the Clover Biopharma in collaboration with the GSK is preparing protein subunit vaccine, and the University of Queensland, Australia, they are trying to prepare this vaccine. Now, why I'm giving you these examples are these are very important trials which are almost reaching phase 3. So I'm giving you these examples. So the one more platform of vaccine is RNA and DNA vaccines. That means you are directly using either the DNA of the virus or a part of DNA or a part of RNA of the virus. Now this is something which is more recent. That means the RNA and DNA based vaccines. That means you are injecting the viral RNA which is modified into the human body and seeing its effect. Now these are RNA viruses. The Moderna and the Pfizer companies, are preparing RNA vaccines based on RNA technology. Now, the last one, which is important, is viral vectors. Viral vectors means, see, in the first case, you are giving the same virus which is inactivated, dead. Second case, the same virus which is alive but weak. The third case, a part of virus which is protein subunit. Fourth case, a part of virus which is DNA or RNA. Fifth one is you are giving a weaker version of the same virus. Like for example, coronavirus is a adenovirus is a cold and normal flu virus. We all get cold and that is because of a flu virus, which is adenovirus. Now that virus is not harmful. We'll just at the max get a cold. Now what they do is they pick up this adenovirus and add some components of coronavirus in it. Now these are known as viral vector based because here adenovirus is acting as a vector. This is the fifth type. The Sputnik V, which is being prepared by the Gamaleya as well as the Russian defense research, is based on this viral vector technology. We are using a weak adenovirus to vaccinate us. So there are various strategies of preparing virus, and the timeline of virus is also important. But finally, before ending my talk, I think I have more 5 to 10 minutes, sir.
Yes, sir.
Yeah, 5 to 10 minutes. I'll just complete it off.
Okay, sir.
Not able to move my slides, just a second. Can you see my slides?
Yes, I am.
But I'm not able to move my slides. Just give me a minute. Just a second. Can you see the screen now?
Yes, sir. Yes, sir. Go ahead.
Okay, sorry for the interruption. so, so finally, vaccines are not so easy to prepare, and there are various challenges which are general in nature and specific in nature. Like, what are the challenges which are being realized now when preparing a vaccine for coronavirus? Some I am trying to put it out. One is virus is automatically very mutative in nature. That means it is highly mutating. The coronavirus 2, which has infected us, has an aspartic residue in its spike protein. But recently they have isolated another strain from a human, which is now mutated to glycine. Now, aspartic acid is a charged amino acid and glycine is a neutral amino acid. So there is some mutation in the spike protein, which is very important for binding. So we really don't know what these kind of mutations can do. Say if you are preparing a vaccine for a strain which has aspartic acid, and the virus cleverly mutates itself into a vaccine which is a protein which is having glycine, and the structure of it can change, and it can again easily enter the cell. So the whole preparation of this vaccine and the whole effort will be wasted. So this is one particular problem. And the second problem is preclinical issues. Sometimes when you inject into animals, they might not elicit antibodies. That means if you are injecting this tested vaccine into a mice or a chimpanzee, it might not actually give you, it might not be a good model. That means it might not elicit antibodies. So, and again, therefore, what they are using is they are using a model known as ferret model. Ferret is a kind of mammal which is very much sensitive to viruses, especially cold viruses. So it can actually elicit a very good immune response against the vaccine, tested vaccine. I mean, candidate vaccine. So this is one of the issues. You have some preclinical issues, you have mutational issues, you have ADE hypothesis. ADE hypothesis is which is very much coming up in case of coronavirus. This is antibody dependent enhancement. That means sometimes what we expect is when you actually inject vaccine, the antibodies would be elicited, and the antibodies should actually bind the virus and clear the virus. That is what we expect. But what happens in case of ADE hypothesis, the antibodies which are produced against the virus will not clear the virus. That means they are not very much specific to virus, or they are produced in suboptimal concentrations. And instead of clearing the virus, what they do is they just go and bind to the virus. And when they go and bind to the virus is treated as self. So what is happening here is sometimes developing of wrong antibodies. If you imagine that the wrong antibody goes and bind to the virus, and that antibody is yours. So that antibody is now bound to the virus, and the virus and antibody both will be happily taken up in the cell. In this case, the virus will be more replicating than the normal. That means if you are giving a vaccine, the person might die faster. So this is one particular fear of the vaccine. Although scientists are very much aware of all these things and they are taking adequate necessary precautions to do so. When you're preparing protein-based vaccines, there are issues of culturing them in larger amounts and fermentation technology and all. So protein-based vaccines are not really tested in a big way, but DNA and RNA-based vaccines are newer platforms. But again, there is a problem with DNA and RNA-based vaccines is these are very new technologies. And till date, there is no RNA-based vaccine. So that means we can understand, although people are trying to make it, there is no registered RNA-based vaccine so far. And the fifth issue, which is very important, is short-lived immunity. Okay, today you have injected the vaccine and you are getting antibodies. But what happens if the antibodies are short-lived? That means, okay, today you have coronavirus infected, and therefore they will the antibodies will be clearing. But the next time, if the coronavirus is again infecting the same person, whether the antibodies are eliciting. Initially, whatever antibodies which are elicited in our body are IgM, and the second antibodies which are released are IgG. Now, IgG are having the memory. Memory means it will have the memory that, okay, the virus is there and I can actually attack it the next time it attacks. So the antibodies should not be short-lived, but they have to have the memory also. Now, the further problem is infection versus transmission. It happened in one of the vaccines, the Oxford vaccine. They have made a vaccine and they have tested it in case of certain individuals. Everything was fine. The lung got cleared up and the virus is eliminated. The virus is not growing into the lung. But the virus is attached to the nasal surface. That means the virus cannot infect the person, but the virus can spread. See, the virus is not in his lungs. He is not infected with the virus, but the virus is absorbing onto the surface of the epithelial cells. That means in that case, if he sneezes or if he is under contact droplet infection, he can transmit this virus. He need not be infected, but he can just transmit the virus. So that is again a big challenge in case of coronavirus vaccines. And there are various other responses like ORF3b, which also blocks these interferons. There is a protein in case of coronavirus, which is known as ORF3b, which is blocking interferon gamma and interleukin 2. Now, these are again inflammation molecules which are important. So there are various problems at various stages when you are preparing a vaccine and when you are administering it into various candidates. The current Sputnik V, which is being registered, is just registered, which cannot be as on date is not released. It is currently undergoing phase 3 trials, and it might take at least 3 to 4 months to undergo those 3. Until unless phase 3 is cleared, FDA would not give approvals for its release into the international market. So whatever testing would they be doing, will be doing it in Russia. As far as the reports say that they would only do it till December, and if at all they are providing successful results till December, then it might get into the market by January. So I mean, like it's very premature to jump into conclusions, and so I request all key we have to be still cautious for till an efficient vaccine is developed from whichever country. It should be for the welfare of the humankind. Now, these are the list of various vaccines which are under phase 3 trials. More you can see that the University of Oxford, AstraZeneca, Sinovac, and you also have Bharat Biotech from India in the race, Codagenics, Serum Institute of India, Griffith University. These 3 are in phase 2 trials. Now, these above are in phase. Totally, there are approximately 28 candidates in phase 2, phase 3 trials, and there are more than 100 vaccine candidates in case of preclinical trials. So this is about a short story. Actually, I mean, like there are so many things which are coming up in public domain every day. There are thousands and hundreds of research articles which are being published. So to give an overview for the student, catering to the student of undergraduate and postgraduate student, I think this information would be suffice. So I sincerely thank all the organizers of this and appreciate the kind of initiative they have taken, especially Sri Polisetti, Sri Srinivas Babu, Sri Ravinder, HODs, Department of Biotechnology, Zoology and Botany. And I also thank my parent university, Central University of Punjab, for always encouraging me. And I also thank all the students and the other faculty which are attending this webinar. So at the end, I think the take-home message is there will be light at the end of the tunnel. Keep faith in science and scientists. Stay safe till then and follow all the government norms responsibly. Hopefully, there will be a vaccine very soon. Thank you so much, one and all, for your patience. So I am done, sir.
Thank you, sir.